Presentation, I'm going to be explaining the concept of linked genes. Now, the syllabus point says that gene loci are said to be linked if they are on the same chromosome. And you can see here in this diagram that genes A and B are linked because they're on the same chromosome, whereas genes A and C and B and C are not linked. So what we've looked at so far is actually unlinked genes. We've looked at if we had, say, B and C. But what we're going to be looking at in today's PowerPoint is what happens when those genes are actually linked and are on the same chromosome, like A and B. Okay, so this was discovered in 1905 by three scientists called Bateson, Sanders and Punnett. Yes, he was the same person who actually developed the Punnett square. And they were looking at doing some breeding experiments with sweet peas, and they were looking at two different genes, one for flower colour, and flowers could either be purple or red, and the other gene looking at pollen grain shape, which could either be long or short. And they knew from breeding experiments when they bred them together that the purple flower was dominant over the red flower, and the long pollen was dominant over the short pollen. Okay, so a cross was made between two plants that were heterozygous for both genes. So what was the phenotype of the parent plants if they were both heterozygous? Well, if they were both heterozygous, they both had a dominant purple allele, and they both had a dominant long pollen allele. So all of these plants were actually purple flowered and long pollen. And the genotype of the parent plants, well, we'll look at notation, proper notation for linked genes later, but we know that because they're heterozygous, they would have had this genotype for flower colour, and they would have had this genotype for pollen. Now, if we were to do a normal cross between two of these, so we do a cross between two heterozygous, so we cross this with this, we would have expected a particular ratio, and we would have expected for dominant and dominant, you'd have expected 9, and for dominant with recessive, you'd expect 3. For recessive with dominant, we would expect 3, and for recessive with recessive, we would have expected 1. So that's a ratio they would have expected. And they actually bred loads and loads of these plants together, and they got a large number of offspring, and the actual numbers that they got were these. So in fact, they bred in total 2,132 plants in total. So that's a really large number, so they can be, quite happy with their results. And they did this over time, and they got these results. And this didn't follow the normal ratios. We would have expected a ratio of 9, 3, 3, 1 here. But in fact, what they got were these numbers, which didn't actually follow. So the numbers for 9331 would have been more like, say, 1,200 here, 400 here, and about 133 here. Now, you can see here and here, it's not that far off. I mean, that could be down to chance. But the fact that you've got like almost a third less here and here compared to here shows that something's not going right. And this breeding experiment did not follow Mendel's law of independent assortment. And they could be pretty sure about this because they had such a large number of offspring that they produced from these breeding experiments. So, what does this mean? So this just shows how that they bred the initial parents. They produced those heterozygous offspring, and then they bred them again through self-fertilisation, and they didn't get the ratios that they expected. So why don't linked genes follow the normal rules of independent assortment? Well, if we look very basically at what we were looking at to begin with, if you've got in an individual a pair of homologous chromosomes, and this individual is heterozygous, so you'll have a dominant one here and a recessive one here. The other gene is also on the same chromosome, like this. So when these two chromosomes separate in meiosis, you'll have gametes like this, and you'll have gametes like this. You won't get so many of the dominant with recessive, so dominant A with recessive B, or dominant B with recessive A. You won't get as many of that combination. Now, this can happen through crossing over, but you'll get far fewer. So if we look at this one here, you can see that occasionally crossing over will happen between the two genes. And in this case, you would get 4 different possible gametes. You'd have that, you'd have this, you'd have this, and you'd also have the 2 recessive together. But that's going to happen not all the time. A lot of the time you will just get these 2 being produced. So you're going to get a lot more of these 2 gametes than you will of these 2 gametes. There's going to be a lot fewer. You're not going to get even numbers like you would if A and B were on different chromosomes. Okay, so that's how recombinants are formed. Now, finally, I just want to talk a little bit about notation. Now, if we were looking at a heterozygous parent, we would normally, if we're just doing unlinked genes, we would write the notation like this, which is absolutely fine. And then you would do the gametes that look like this, and this, and this, and this. And that would be absolutely fine to do it like that. However, with linked genes, you actually show them on the same chromosome. So in this case, you would show the dominant was linked like that, and then the recessives were linked on the other homologous chromosome, and you do little lines. And in this case, when you show the gametes being produced, you actually leave the gametes on the same chromosome like that, and then this one would be on that chromosome. And then to show the formation of recombinants, you actually show a little chiasmata being formed between the 2 chromosomes, and then you can see they've crossed over like this. And then, so you can see that this pair here have actually swapped over. And then we can show that those gametes will form like this. And then, okay, so we get more of these forming because crossing over doesn't need to take place for those to happen, and we get fewer of these, so we get far less of these particular gametes. Now, the number of these gametes that we get depends on how close those genes are together on the same chromosome. So if they're quite far apart, like on this one, crossing over is going to happen quite a bit between these 2. So you're going to get more of these, but not as many as these and these. However, if the genes are very close together, if the gene B was sort of up here, crossing over would be a lot less likely to happen. And in that case, you would get very, very few of what we call these recombinants being formed here and here. So that is how linked genes works, and that's the notation. In the next lesson, I'll talk a little bit more about some breeding experiments that were done by Thomas Hunt Morgan on fruit flies, and also how we can test some of these theories using chi-squared.